Okay, in 18-2, we're going to actually talk about different hormones and the different types of hormones and how they talk to cells and tell them what to do. So first, let's start with the organs of the endocrine system that are making these hormones. The goals of the endocrine system are to promote growth and development, reproduction, regulate cell metabolism and energy balance, regulate water balance, electrolytes and organic nutrients, and then mobilizing body defenses. We also need to differentiate the difference between endocrine and exocrine. So endo means within, exo means outside of. And so exocrine hormones will be secreted into ducts, which will be leaving the tissues, versus endocrine or an endocrine release would be something that would be deposited into the bloodstream or into an extracellular fluid of some sort. Here is a picture from your book that goes through all of the hormones and the organs of the endocrine system and the hormones that each of them will release. But I just want to pause for a second and take us to the Complete Anatomy app to kind of look at what this looks like for the entire body. So right now we have the lymphatic system highlighted, but let's turn on the endocrine system here just so we can identify what's going on. So this is a male skeleton, and here we could see the testes at the bottom, which are going to be an endocrine organ, the pancreas in the abdomen, just inferior to the stomach, the adrenal glands, which are going to sit on top of the kidneys in the abdomen, and the thyroid gland, which is going to be in the cervical region. And as we can imagine, all of those will be, there's also the pituitary gland, which isn't, you can't really see it from this view here, but let me turn on the arterial supply. And as you can imagine, there's a lot of arterial supply throughout the entire body, and these organs are very close in close contact with the arterial supply, so that once they make those hormones, they have an easy method to deposit them somewhere and send them on their way. Cool. Okay, so we have two types of we have three types of classes of hormones. The first category, which we won't go into too much detail of in this class, but we're just going to introduce it here, are amino acid derivative. So if you remember amino acids, these are the monomers of proteins, and you can essentially imagine a handful of amino acids coming together and changing in their conformation or changing in their shape or their size, and then that actually acts as a hormone because it can initiate some sort of chemical signal within some sort of tissue. So some examples are thyroid hormone, epinephrine, norepinephrine, dopamine, and melatonin. What we're really focused on in this class are peptide hormones and lipid derivative hormones. So first starting with peptide hormones. Remember peptides, these are proteins. These are polymers of amino acids put together, and they will create some sort of quaternary structure, and that quaternary structure will be able to interact with other receptors or other molecules on a cell surface in order to initiate some sort of response within that cell. So some examples of how they would interact with the cell membrane: they could interact with glycolipids or glycoproteins and small polypeptides. And this is really important to understand because we'll talk about how peptide hormones can interact with a cell and tell the cell what to do. And in order for the cell to register that a peptide hormone is there, it actually has to bind to some sort of receptor in order to initiate that response. Versus lipid derivatives are different, right? So lipids, we've talked about lipids from 201, and it should be a reminder from Bio 111. But lipids are another class of polymers in the body that we use, and they are the components of almost everything. You'll see lipids everywhere, including the plasma membrane, like in this picture. And because of that, lipid hormones don't need some sort of specialized receptor in order for them to tell the cell what to do. They can actually just kind of like squeegee their way into the plasma membrane or diffuse across the plasma membrane, because like equals like, right? They'll work with like type molecules. So we don't need any sort of specialized receptor in order for the lipid hormones to do what they need to do. They can also be used as signaling molecules within the bloodstream. So some examples are leukotrienes, prostaglandins, thromboxanes, prostacyclins. We won't talk about the three, but we might hear about prostaglandins a couple times in this class. They're named prostaglandins because they were originally found in the prostate, but they were originally discovered in the prostate of males. But we actually have seen prostaglandins everywhere throughout the human body. They're usually a pro-inflammatory type of molecule, and but the name has just stuck around, so don't let it confuse you. It's not just about the prostate. The pictures on the right are very, very complicated. Don't worry about memorizing this at all. In fact, if it confuses you, don't even worry about it at all. All I'm trying to show you is that lipids are really great at becoming other lipids. So only if you add or tweak a part of the molecule, even ever so slightly, it can completely change into a completely different hormone, which is really important because this makes this process a lot more simple and efficient if we're trying to communicate with other parts of the body. So, there's only a handful of differences between estrogen and progesterone, even though they're both used in women. But estrogen is still also used in males too, so estrogen is only a couple different modifications away from being testosterone. And all three of those are actually derivatives of cholesterol. So we actually always start with cholesterol, which is made from arachidonic acid, so that's why in the yellow box there you see arachidonate, I don't know how to pronounce that, arachidonate, I don't know. But arachidonic acid creates cholesterol, and then cholesterol turns into all these other types of lipid hormones. So that's what that picture is trying to show you, is that you can have all of those derivatives just off of cholesterol. In addition to cholesterol, we also have steroid hormones. So they look kind of like cholesterol, but they have a couple extra 6-carbon membrane rings attached to them. So similar to cholesterol, but that's like a subcategory of a lipid type of hormone. And here I have C figure 8-2. Okay, so let's talk about how, in general, these might be floating around in the blood and then bumping up into their right tissues. So we can imagine that they are floating around in the bloodstream at any given time. And if they're a hydrophilic messenger, like in picture A, hydrophilic, right? So this is water-loving. And they like water, so they can easily float in between the cell and the blood vessel into within that extracellular fluid. Versus hydrophobic messengers, they might actually need some sort of carrier protein in order for them to get from point A to point B. Because they're not comfortable floating around in the water by themselves. They need to be protected by something. So all we're trying to say here is that it really depends on if you're a peptide hormone and you can float around freely, or if you're a lipid hormone and you are trying to find your active cell, or the cell that you're trying to activate. And you might not be able to float around very freely because you're made out of lipids, and lipids don't like water. They like to stay away from water, so they might need to carry our protein. So in general, whenever we have the hormone bind to its receptor, wherever that receptor may be, there's one of three things that can happen after it binds. We can either immediately go to the nucleus and bind to the DNA and initiate transcription or turn off transcription, or choose whatever genes we want from the DNA in order to transcribe them. So this can all happen in the nucleus. Or we can alter the rate of protein synthesis. So this would be modifying the ribosomes. Remember, ribosomes can be free-floating in the cytoplasm, or they can be attached to the endoplasmic reticulum as part of the rough endoplasmic reticulum, or the rough ER. And when we alter the rate of protein synthesis, maybe once we bind, we're increasing protein synthesis, or maybe when we bind, we decrease protein synthesis, just depending on whatever it is that's bound to it. We can also alter membrane permeability. So once we bind to the membrane, maybe the membrane then becomes less interested in opening up its ion channels for a specific ion. Or maybe it becomes more interested in opening up an ion channel to allow ion either in or out of the cell. So these are one of the three ways that after a hormone binds, it can change the inside of the cell, or change the way the cell is actually operating. And we'll talk about some examples of how this happens and why it's important here in a second. So in general, we have the hormone, it binds to its specific receptor, and it initiates some sort of downstream effect, right? And we can either, if we wanted to propagate this signal, if we wanted to have more of an amplification of this signal, we have a couple ways that we can do this. Either we can dump more hormone in, but there's only so many receptors. So we really need to have as many receptors as there is hormone in order to really upregulate the signal. So really we need to upregulate the number of receptors in order to increase the potential for the signal to, or in order to amplify the signal. We can also downregulate the receptors. So if we get rid of receptors, but we have the same amount of X hormone being made, then now we're automatically getting rid of the amplification. So this is why I have why alter the receptor and not the hormone, right? Like if you go into a buffet and you are the, let's say, yeah, you're the hormone, right? Or you're the receptor, excuse me. You're the receptor and you go into the buffet and you're trying to eat everything in the buffet, right? And if you're the receptor and you're going to eat everything from the buffet, and let's say everything from the buffet is those hormones, right? You're going to get full at some point, right? You're going to max out at some point, right? You're not going to be able to eat everything that's at the buffet. So in order to eat everything that's at the buffet, you recruit some of your friends, you recruit some more receptors, you bring more of the receptors to the buffet, they're able to actually eat everything at the buffet, and then you walk out, you all roll each other out of the buffet eventually because it's too much food. So if that analogy helps you, awesome. But that's the whole point of why we care about what's happening with the receptor as well. We don't just care about what's happening with the hormone. All right, so let's talk a little bit more in detail about how these receptors work, and in one receptor in particular, which is called a GPCR. So I'm just going to close that off to the left for a little bit so we can just talk about what a GPCR is. So a GPCR. So here we're only talking about peptide hormones. Okay. And remember, peptide hormones, these are proteins. They're going to need a receptor on the outside of the cell to initiate some sort of response, because they can't just like pass their way through the plasma membrane willy-nilly and then just get to their target inside the cell. They need to have some sort of receptor. So we're going to use a GPCR for that, and GPCR stands for G, protein, coupled, receptor. Okay, so we're working with a G protein. That G protein is going to work after it is activated by the protein at the receptor. Okay, so we have a couple different types of GPCRs. We have some that inhibit cell activity, and we're going to call these G alpha I. We have some that stimulate. We're going to call those G alpha S. And we have some that increase intracellular calcium levels. We're going to call those G alpha Q. Okay. Alexander, do I really have to know these letters? No, but I think it really helps to kind of break this apart into the three different methods that these GPCRs can work. So we're never going to ask, describe what a G alpha I does. No, we're going to talk about some component of that cascade, and we're going to expect you to know it. But while you're learning it, I think it's important to kind of classify them into their names so that you can use them while you're studying. Okay, so GPCRs in general, right? Let's say we have a plasma membrane. What do they even look like? We have a plasma membrane. And on the outside of the plasma membrane, we're going to have this receptor. And this receptor is special. It goes through the membrane once, twice, 3, 4, 5, 6, 7, right? 1, 2, 3, 4, 5, 6, 7. 7 times. And then it ends on the inside of the cell. Okay? And this is important because once something binds to the outside of the cell, the receptor of the cell, it induces some sort of conformational change to the receptor itself, and then the downstream effect is actually on the inside of the cell, which is what we want. We want to initiate some sort of response on the inside of the cell. So these are the GPCRs, okay? And all the GPCRs are going to have the same they're all going to have the same receptor, except what happens at the end of the receptor is going to be different between I, S, and Q. Okay, so we're going to talk about those 3 examples next. Okay, so let's go ahead and start with G alpha S. So I'm going to draw one cell. And, yes. And the plasma membrane. Okay. Oh, I guess it wants to... Oh, goodness. Oh my gosh, go away. Go away. There we go. Okay. And this is going to be G alpha S, so stimulatory. So stimulatory, right, we're going to increase the activation of the cell. The cell is going to increase what it's supposed to be doing. And if you're following along with the pictures on the left, we're starting with the first picture on the left. Okay, so for G alpha S, let's zoom in here for a second and let's draw that GPCR, because we know that we're always going to start with that GPCR. 1, 2, 3, 4, 5, 6, 7. Okay, so we are waiting for a hormone to bind. A peptide hormone binds. Peptide hormone binds. That's step number 1. Okay. After the peptide hormone binds, we have to do some things on the inside of the cell in order to promote further activity, right? So at the end of the G protein, we are actually going to have a ADP molecule. Once it's activated, gets changed into an ATP molecule. And we like ATP, right? ATP stimulates things, turns things on, makes things happen, right? And ATP is going to excuse me, let me back up one second here for a second, sorry. So this right here, this is going to be our G protein. I need to identify that. So we activate the G protein then turns ADP into ATP. Then ATP will go and turn on a enzyme called let me make sure I spelled this correctly. Adenylate. Oh, yep, see, I already misspelled it. Adenylate cyclase. Okay, so we know it's an enzyme because it ends in ACE, right? And adenylate cyclase is very good at stimulating CAMP to turn on. So adenylate cyclase is going to go and activate, so I'm going to draw this, CAMP, cyclic AMP. Okay, and we're going to activate CAMP. And once we activate CAMP, then CAMP can do a couple of things. It can do number 1 or number 2. Number 1 is it might open up some ion channels. Right? And this makes sense because we said one of the 3 things that these hormones can do once they bind their receptor is change the membrane permeability. And so that's how we would be doing it, is by opening up ion channels. Or CAMP can turn on other active enzymes, or can either activate other enzymes. Okay, so that's nice because then after we activate other enzymes, then that might stimulate another response and another response and another response. And so we call this like whole, like stepwise thing, a cascade of events. Right, so we stimulate some sort of cascade response within the cell. I also want to mention that once CAMP gets turned on, it activates kinases. Kinases are a protein type of enzyme that are used to turn things on in the cell. So this is all G alpha S, right? Peptide hormone binds. And actually, let me highlight like this. Peptide hormone binds, that's step number 1. It activates the G protein on the inside. The G protein converts ADP into ATP. ATP then goes and turns on adenylate cyclase. Adenylate cyclase goes and activates CAMP, or cyclic AMP. Cyclic AMP turns on kinases, and those kinases are able to either open up ion channels at the membrane to change the membrane potential, or the membrane permeability, or they can go and activate other enzymes to stimulate some other downstream response. All right, so that's all G alpha S. Okay, let's do the same. You can always pause, rewind, repeat this video. Let's go ahead and move on to G alpha I next. So as you can imagine, G alpha I is going to be really similar to G alpha S, except we're going to turn things off. Okay, we're going to turn things off. And let's go ahead and start at the plasma membrane, where we have our G protein that spans the membrane 7 times. And we wait for the hormone, peptide hormone, to bind. That's step number 1. Peptide hormone binds, turns on the G protein. The G protein then turns on, the G protein then turns on an enzyme called phosphodiesterase. Phosphodiesterase. You know, it's an enzyme because it ends in ase. Phosphodiesterase then gets activated, and phosphodiesterase will go and talk to CAMP, cyclic AMP, and it will make it inactive. And cyclic AMP will then no longer produce enzyme activity. So we're decreasing enzyme activity. Okay. All right. So step number 1, peptide hormone comes and binds, activates the G protein. G protein activates phosphodiesterase. Phosphodiesterase turns off CAMP and therefore decreases enzyme activity. Okay, perfect. So why do we even talk about these GPCRs before we move on to G-alpha-Q? GPCRs are one of the most abundant type of peptide hormone receptors in the body, and they are, I think it's like somewhere between like 80 and 90%. Like they are the vast majority of the receptors in the body. And this is really useful to know, because that means we can target a lot of different drugs to target these parts of the cascade, right? So we can either, if we wanted to go back to G-alpha-S, right, we could stimulate some sort of response to ruin the receptor here, or maybe we could ruin the G protein here. Maybe when we want to still increase ATP levels, so maybe we break something off of the downstream effects, right? So like, you know, if someone has some sort of disorder that is prone to turning on one of these enzymes and making it overactive, right? We could diminish that response by working somewhere upstream of the cascade in order to turn it off. So that's how a lot of drugs actually work, is they target some portion of this cascade, depending on what type of receptor it is, to make a person healthier. Okay, so let's finish with G-alpha-Q. And G-alpha-Q is probably the most complicated one, because there's two things happening at the same time. So we're going to start with the first thing that's happening, G-alpha-Q. And the first thing that's happening, as with all of them, is the G protein. 1, 2, 3, 4, 5, 6, 7, good. Okay, receptor binds. Receptor binds. And the G protein does one of two things. We're going to talk about the first thing, and that's, I'm talking about image B over here. So the G protein gets activated and goes and stimulates the release of intracellular calcium. And where's calcium stored? From the endoplasmic reticulum or the sacroplasmic reticulum. Okay. What's the point of having a bunch of calcium? Well, calcium can do a lot of signaling things. Calcium is also very good at binding onto things and turning things on as well, just like kinases would. And so calcium can float around in the cell, and once we have a lot of calcium, it can go and bind to an inactive protein called calmodulin. And once it binds to calmodulin, and we have calcium bound to it, then that makes it active, and calmodulin can go and then stimulate the activation of other enzymes. Okay, but honestly, this response is not efficient, right? Because we only have so much calcium in the intracellular stores, so we have to get calcium from somewhere else. So in order to do that, we're going to have to open up a calcium ion channel from the membrane to allow calcium to flow into the cell. So that we have more calcium accessible in the cell to activate calmodulin. So how do we do that? let's go back to the first step. And in the first step, we activate the G protein. And the G protein is actually going to go back to the membrane and change in something in the membrane called PIP3. This is going to go and activate PIP3. And PIP3 is going to turn into one of two things. Not into one of two things, but it'll break apart into IP3. So just rearranged itself from PIP3 to IP3. And then it also left behind a piece called DAG. Oh, I should probably draw that bigger. Sorry. DAG. And I'm just drawing arbitrary shapes. That, I mean, who cares about the shape? I'm just drawing them. So PIP3 separates itself into DAG and IP3. Okay, we don't care about DAG anymore. We care about IP3 because IP3 is going to take us through the rest of the cycle. So IP3 is then activated, and IP3 can then go to the to another enzyme called phosphokinase C. Phosphokinase C. And then phosphokinase C gets activated, and phosphokinase C can then go and activate the opening of the calcium ion channels, and then calcium flows into the cell. Calcium can then go and bind to calmodulin, and then we can continue this downstream effect. Okay, let me start that over from the beginning. All right, so peptide hormone binds to the G protein coupled receptor. The G protein at the end of the receptor does one of two things at the same time. This is happening at the same time. One is it converts PIP3 into IP3 and DAG. The other is that it releases calcium storage from the inside of the cell, either from the endoplasmic reticulum or the sacroplasmic reticulum. And we have free-floating calcium done on the left side. Then after IP3 gets activated, it turns on phosphokinase C, and phosphokinase C recruits even more calcium into the cell. And once we finally have all of this calcium into the cell, we can finally turn on calmodulin by binding to it, and then we finally have an increased cellular response. Okay, I know that was a lot. I would just do it multiple times. This took me quite a bit to get it in my head as well, and you see me even take a second to go back and add some things. So I would recommend drawing this out many times until it's memory for you. Okay, so now we talked about how peptide hormones work. Let's talk about how steroid hormones work. Okay, so steroid hormones. And draw a cell, plasma membrane. Okay, so instead of having a GPCR, we don't even need a GPCR, because instead, these steroid hormones, and let me do this in a different color, I suppose, these steroid hormones can just pass through the plasma membrane, no problem. Because like equals like, they're made out of lipids, the phospholipid bilayer is made out of lipids, so they can just squeegee their way past the phospholipid bilayer. And once they do that, they have one of two things that they can do. The first is that they're going to go and find their receptor. And their receptor is in one of two places. Either their receptor, and I'm just going to draw it as an R here, is floating in the cytoplasm, or their receptor is in the nucleus. Okay, so either the steroid hormone goes and binds to the cytoplasmic receptor, or it goes and it binds and passes through the nuclear membrane, and then binds to its receptor in the nucleus. If it binds to the cell receptor in the cytoplasm, then it will be carried into the nucleus anyway, and that is where it'll become activated. So once it's brought into the nucleus, it's activated. And once it's activated, it can go and talk to DNA, or actually directly bind onto DNA. And once it binds onto DNA, DNA becomes active, or activated, and transcription happens. And remember, transcription is where we go from DNA to RNA. And RNA can then leave the membrane and become translated in the cytoplasm by ribosomes. And once it's translated and packaged by ribosomes, then it can either stay inside the cell and do what it needs to do, or it can leave the cell and do what it needs to do. The other thing that can also happen is that steroid hormones can also come and bind to mitochondria. So I'm just going to abbreviate mito here. So they can also come directly and bind to mitochondria, and if they bind to mitochondria, then that's going to increase ATP production. And that's it for steroid hormones. Relatively simple, right? All we're doing is we're coming into the plasma membrane, just squeegeeing our way through, and we have one of three ways we can get there. We can either go to the mitochondria and make ATP, we can bind to our receptor outside the cytoplasm and come into the nucleus, or we can find our receptor in the nucleus, but once we're in the nucleus, we're activated. Then we go to the DNA, we transcribe, or we turn on the DNA, we latch ourselves on directly onto the DNA to initiate transcription, and then the mRNA leaves the nucleus to become translated by ribosomes and packaged by ribosomes, and then it leaves the cell and does whatever it needs to do. That's it for steroid hormones. So make sure you understand the difference. There's a big difference between how we activate or how we use peptide hormones via G-alpha I, S, or Q, and then we use steroid hormones in a completely different way. Okay, so steroid hormones, this is also why it takes longer, because the GPCR can immediately initiate this activation, right? The peptide hormone can come, find its receptor, bind, and then we have this very, very quick cascade. Even though there's a lot of steps in this process, it's very clearly driven, and everything has a very clear endpoint, so it happens really, really quickly. Versus steroid hormones, I mean, it kind of takes a long time for the peptide hormone to finally pass its way through the membrane. Then it has to find its receptor, right? So maybe it'll come and dance over here, or maybe it'll come and dance over here, and then it's like, oh, look, then there's my receptor. So that takes some time. Then it has to get its way into the nucleus again, which that also takes some time. Then we have to transcribe, which takes some time. Then we have to translate, that takes some time, and then we finally leave the cell. So this is why steroid hormones always take a much longer time to actually have some sort of effect. Versus peptide hormones, they bind, and then boom, 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 things happen. So that's also really important to understand. Okay, up next is we need to talk about the 3 different ways that we can actually control the types of secretion of these hormones. Okay. The first way is hormonal. The second way is humoral. And the third way is neural. Okay, what's the difference? All right. Okay, so hormonal means that we're making hormones and we dump them into the bloodstream, and then they go and they find another organ, and then that organ makes more hormones. Versus humoral, humor means like a body of fluid, and so we're talking about blood here, and so blood is floating around at any given time, and then depending on the composition of the blood, that will activate some sort of nearby organ to induce whatever it needs to do to get us back to homeostasis. So maybe it secretes some sort of hormone, and then that gets us back to homeostasis. And then neural, right, the nervous system has to be stimulated, and then the nervous system goes and talks to an endocrine organ and tells that organ to turn on and make hormones. Okay, so let's go ahead and write all of that out. So hormonal, right? We are going to give an example which is provided in this picture too. We have the pituitary gland, which we'll talk about in the next section. The pituitary gland is going to make some hormones, and it's going to spit out those hormones into the bloodstream. And then those hormones are going to float around in the bloodstream, and then they're going to go and find their target organ. Oh my gosh, if I could spell organ. Target organ. And that target organ could be one of 3 things. One being the thyroid. One being the adrenal cortex. Cortex CX. or the gonads, either testes or ovaries. And then all three of those are going to make some sort of hormone. And then those hormones are going to go to another tissue. And then that's going to stimulate the wanted response. Phew, that was a long process, right? Okay. Versus humoral, right? We are floating around in the blood. And the blood is going to have some sort of composition, right? And we're going to float past some sort of nearby organ that can senses a disturbance in the force, senses imbalance of whatever it is. Either there's too much CO2, or there's too much sodium, or it's really acidic, or it's really basic, whatever that is. And then that organ is going to secrete a hormone to counteract that. And then that hormone is going to go into the bloodstream. And then that's going to induce whatever the wanted response is to get us back to homeostasis. Okay. And then the neural response, the easiest one, I think, found of the three of them. The central nervous system gets activated for whatever reason. And that stimulates its axons to talk to target organs. And then that goes and they secrete a hormone into the bloodstream. And then that induces the wanted response. And the whole intent of all of these wanted responses, right, is that we finally get back to homeostasis. Whew, that was a lot. Okay. Do you really need to know like all of these steps and memorize this backwards and forwards? No. We're going to give you a lot of examples. Like we're going to talk in detail about the thyroid, the adrenal glands, the gonads. We're going to talk a lot about what part of the blood is that we care about and what that would induce, what type of hormone to be released to get us to that wanted response. And we're going to talk about those specific neural responses. So this is just kind of more of an introduction, but if this doesn't make any sense to you, then definitely focus on this slide before moving on. Okay, we'll see you for the next section.